Immunity protects us from cancer and infectious diseases. It's essential for our existence, and so fundamental that we share much of it with primitive animals, such as sponges. Adaptive immunity evolved more recently, but still requires an innate response to be initiated. Natural killer, or NK cells, are the major cytotoxic lymphocyte within our innate immune system. They constantly patrol our bodies to eradicate tumor cells as they arise and provide crosstalk to the adaptive immune system. Failure of an NK cell to recognize a tumor cell allows cancer to develop. Understanding tumor-specific NK cell responses is an important step towards maximizing the therapeutic potential of NK cells in cancer treatment. The tumor surveillance by NK cells in healthy individuals provides a rapid response to tumor formation and is vital for the early response to tumorigenesis. NK cells are particularly important as, unlike T cells, they can kill tumor cells in the absence of antigen-presenting cells. So, while resting NK cells circulate in the blood, their activation is controlled by a multi-step signaling process requiring multiple ligations of molecules expressed on their surface. This cascade of signals moves the rest to the point where they can kill tumor cells. Most tumor cells provide enough of these signals to drive resting NK cells through to release of lytic granules and cytokines. But tumors evolve, and genetic changes can offer them a survival advantage, as losing expression of the molecules that the NK cells recognize. These NK-resistant tumor cells lack sufficient ligands to activate NK cells. The resistant tumor cells become more prevalent, and a growing tumor ignored by NK cells can lead to the development of cancer. However, lysis of this NK-resistant tumor can be achieved if a source of additional signals is provided. This can be achieved by priming with an activating cytokine, or more effectively by conjugation to INKmune, a tumor cell which expresses NK priming ligands. Our research at InMune focuses on the signals needed to transition NK cells from rest to lytic function, and on why some tumors are more resistant to NK killing. We've shown that it's possible to overcome the loss of activation receptor expression in NK cells by priming them with INKmune. INKmune cells bind to NK cells via multiple ligand receptors, one of which is CD15. In the NK INKmune synapse, the CD2 receptor on the NK cell binds to CD15 on the INKmune cell, and CD16 is shed. Within the cytoplasm of the NK cell, CD3 zeta released from CD16 then binds to the intracellular domain of CD2 and is phosphorylated. This leads to the downstream phosphorylation of LAT and the subsequent phosphorylation of STAT5. Phosphorylated STAT5 induces IFN gamma synthesis, which leads to the transition from resting NK cell to a primed state. The primed NK cell now needs to bind far fewer activating ligands on the tumor cell to be able to kill it. So cancer cells which have evaded NK cells are now susceptible and are killed.
Our skin, like the bark of a tree, represents valuable protection. However, the skin is also home to countless microorganisms. Among other things, they are nourished and protected by an oily fluid called sebum. Sebum is produced by sebaceous glands. Sebum also contains antimicrobial elements that can kill microbes. If they are harmful or too many microbes on the skin, the sebum is composed in such a way that these microbes are killed. This creates a harmonious coexistence between skin cells and microbes, a kind of symbiosis. These microbes also keep one another in balance and kill pathogens that can be dangerous to our cells. The protective layer of hollow organs, such as the nose, mouth, and trachea, is a mucous membrane. Mucous membranes possess cilia. Cilia move back and forth in a wave-like manner. This allows them to move mucus, to which foreign substances adhere, upward into the oral cavity. mucociliary clearance is not sufficient, an attempt is made to achieve clearance of unwanted particles through coughing. This is done by inhalation followed by a sudden exhalation, so that foreign substances are removed from the lungs and trachea. The bone marrow has stem cells. It is the birthplace for many different immune cells, which we also call leukocytes. Stem cells divide to form daughter cells, asymmetric or symmetric. Symmetric cell division results in either two stem cells or two progenitor cells, as seen here. The progenitor cells divide again and again. Thus, a single stem cell becomes many immune cells or leukocytes. A common immune cell is the neutrophil. Some of the neutrophils remain in the bone marrow, while others make their way into the bloodstream. Our neutrophil belongs to the so-called innate immune system and moves through the blood vessels. During inflammatory processes, the endothelial cells of the blood vessels have special receptors. Thus, initially there is weak adhesion, then strong adhesion, and finally the granulocyte goes through the endothelium. This process is called diapedesis. The path through the connective tissue has already been made by other neutrophils. Our neutrophil only has to follow cytokines, that is, signal substances of other cells, a process called chemotaxis. There they are. Bacteria everywhere. They produce toxins and thus harm the cells of the organism. A neutrophil's job is to eat these bacteria. This eating process is called phagocytosis. In addition, some of the neutrophils produce certain cytokines that attract other granulocytes. Other cells of the innate immune system are macrophages. They arrive on the scene before neutrophils, phagocytose bacteria, and produce a signal substance, tumor necrosis factor. It suppresses the feeling of hunger, causes fever, stimulates leukocyte production. Another leukocyte type is the T cell. T cells possess receptors. What do they need them for? Macrophages are professional antigen-presenting cells. They have the ability to present antigens via certain proteins on their surface, and this initiates certain processes. Some T cells can use their receptors to recognize exactly those antigens of the macrophages for which they have been trained in the thymus. Let us take a closer look at this. T cells are also formed in the bone marrow. They then migrate via the bloodstream to the thymus. Because they mature in the thymus, they are called T for thymus cells. In the thymus, which is one of the primary lymphoid organs, T cells form antigen receptors. Afterwards, they usually migrate to secondary lymphoid organs, such as the lymph nodes and the spleen. In the lymphoid organs, naive T cells and dendritic cells meet. The dendritic cells have already had contact with a pathogen and present the antigen on the surface. They are also professional antigen-presenting cells. The T cell scans the dendritic cell for the antigen for which the T cell was trained in the thymus. If it finds the antigen, T cell activation begins and, among other things, clonal expansion occurs. The cell reproduces itself thousands of times. These cells respond to a specific pathogen or antigen. Then, some T cells start their journey and migrate to other tissues. Once there, the cytotoxic T cells scan other cells with their receptors to look for cells that have the specific antigen on their surface. If they find their antigen in a cell, they know that a pathogen, for example, is in the cell. The cytotoxic T cells begin to induce apoptosis. The cell's debris are eventually eaten by macrophages and new cells form. There are not only cytotoxic T cells, but others as well. T helper cells, memory T cells, regulatory T cells, and others. T helper cells work hand in hand with B cells. B cells possess receptors like T cells. Like T cells, B cells also originate in the bone marrow. They then search for their specific antigen in the lymphatic system or in the bloodstream. If the B cell finds its antigen with its receptors, either activation occurs immediately or the B cell moves into the lymphoid tissue where T helper cells wait. T helper cells, as we've seen before, were also trained for one specific antigen. For this reason, the B cell presents its antigen on the surface. This allows the T cell, with its receptors, to scan the B cell. If it is the antigen for which the T cell has been trained, it gives the starting signal for the division and maturation of the B cell. As with T cells, clonal expansion occurs. Thus, thousands of new B cells or plasmablasts and plasma cells are generated from a single B cell. Memory B cells are also produced, which will spring into action in the event of another subsequent infection. During acute infection, plasmablasts and plasma cells immediately begin to produce antibodies, and they release these antibodies into the bloodstream. Antibodies are also often called immunoglobulins. If the toxin, the bacterium, the antigen is in the blood, the antibody just produced by the plasma cell can bind to it. In this way, for example, a toxin can be rendered harmless and an antigen can be marked so that macrophages can recognize it more easily and phagocytize it more quickly. This process is called opsonization. All immune cells have a stem cell as their origin. It develops into progenitor cells and eventually into the immune cells just shown. Other important immune cells are natural killer cells and platelets.
The immune system possesses a tremendous killing power, which can be used to defend the human body from its own cells that become cancerous. Considering all human bodies develop cancer cells throughout the lifetime, how does the immune system provide such protection? The answer lies in two types of cells. The cells of the immune system that kill cancer cells are CD8 cytotoxic T cells and natural killer cells. First, the CD8 cytotoxic T cells will be discussed. Antigens are proteins that are expressed on the outside of the cells. In the case of a normal, healthy cell, a normal healthy antigen is presented on the outside of all nucleated cells of the body. This normal, healthy antigen is presented by a molecule known as MHC1. Cytotoxic CD8 killer T cells do not kill cells expressing the normal healthy antigen. However, as cells become cancerous, their DNA becomes mutated. This can lead to a mutated or tumor version of the antigen. T cells that have a T cell receptor that match the tumor antigen will become activated by the antigen. In order to activate T cells, dendritic cells can present a different cell's antigen on the MHC2 receptor. CD4 T helper cells can become activated by antigen presentation on MHC2 on dendritic cells. The CD4 T helper cells can then stimulate the proliferation of CD8 cytotoxic T cells. This results in an increase in the number of T cells that can kill cancer cells. When a CD8 T cell binds its receptor to the tumor antigen on the MHC1 on cancer cells, it signals the T cell to kill the cancer cell. The T cell can also kill cancer cells by directly inducing apoptosis. Apoptosis is the death program built into cells and results in the death of the cell. A cell can initiate apoptosis within itself through the intrinsic pathway of apoptosis. However, apoptosis can also be initiated by another cell binding the FAS receptor on the outside of the first cell. This is the extrinsic pathway of apoptosis. T cells present the FAS ligand, which can bind the FAS receptor on cancer cells. Thus, cytotoxic T cells can induce apoptosis in cancer cells by binding their FAS ligands to the FAS receptors on cancer cells, which kills the cancer cells. However, T cells only initiate killing of cancer cells when they detect tumor antigen. It is possible that some cancer cells have DNA that is so mutated, even the MHC1 that presents the antigen cannot be properly built, and no antigen presentation occurs. How is the immune system able to kill cancer cells without antigen presentation? The answer lies with natural killer cells. Natural killer cells are part of the innate or primitive branch of the immune system. This means that they are not specific to one type of antigen, and natural killer cells do not examine the antigen on MHC1. Natural killer cells have two receptors on their cell membranes, the activator receptor, or NKG2D, and a suppressor or inhibitory receptor, or KIR. When cells are under stress, such as genetic mutations that can occur in cancer cells, they produce stress proteins on the outside of their membranes. These stress proteins bind the activator receptor on natural killer cells. Natural killer cells are stimulated to kill when the proteins of a cell bind the activator receptor. However, if the inhibitory receptor is also bound, it will stop the natural killer cell from killing. In order for a natural killer cell to kill, its activator receptor must be bound, and its inhibitory receptor must not be bound. MHC1 is able to bind the inhibitory receptor on natural killer cells. Thus, as a cancerous cell becomes less able to produce MHC1, this means it is less able to bind the inhibitory receptor on natural killer cells. Additionally, cancer cells are greatly under stress. This leads to a large amount of stress proteins on the outside of the membranes of cancer cells. Therefore, cancer cells decrease the amount of MHC1 that would block natural killer cells from killing, and cancer cells increase the number of stress signals that activate natural killer cells to kill. This results in increased activation of natural killer cells, which proceed to kill cancer cells. Natural killer cells kill in similar ways as T cells. by releasing granzyme B or expressing the FAS ligand and inducing apoptosis of the cancer cell, fail to properly produce MHC1 and not be killed by T cells, this same lack of MHC1 makes cancer cells exposed to being killed by natural killer cells. How are tumors sometimes able to evade the immune system and continue to grow? Some cancer cells express the FAS ligand on the outside of their cell membranes. Thus, as T cells and natural killer cells approach the cancer cells, it is the cancer cells that bind the FAS receptors on the T cells or natural killer cells. The cancer cells can kill the immune system cells by the same means the immune system cells would have killed the cancer cells. Additionally, tumors can secrete immunosuppressive cytokines such as interleukin-10 or IL-10 or transforming growth factor beta, TGF-beta, which can prevent the proliferation of immune cells and leave them inactivated. Lastly, tumors can produce large amounts of lactic acid, resulting in an environment too acidic for immune cells to live in. Thus, sometimes the immune system is unable to rid the body of cancer.